Hello, everyone. Welcome to the New Jersey Mycological Association Taxonomy Tuesday session for November 11th, 2025. Happy Veterans Day. We take turns here discussing finds that we made in the past week or so, or the same time a year ago. It's a round robin. We take turns and we share from our computers photographs of the specimens. And we also can show photos that you've emailed into the Taxonomy Tuesday address. So I'll be your host tonight. Again, my name is Lila, and we will get started. Does anyone have anything they would like to show from their device? You can put your name into the chat, and we'll get a queue going there. Or we will go to the email to get things started.
I have one.
All right. Super, Christopher. Would you like to? Oh, here it comes.
I was with my whole family, so, I couldn't really stop for all that long. And the AI here on INAC came up with what was it? The equestre, the Tricholoma equestre. The yellow didn't come up. The smaller one is pretty well yellow under there. Does anybody know these all that well?
It's a species group.
Sometimes they're brown on the cap like this one.
When they get old, I've seen them.
This young one was basically about that same color on the cap.
Yeah, I found some yesterday actually under a mix of pines here in Northeast PA that were brown on the cap. And as Igor said, it's a species complex. So it might be that some years one of the taxa is fruiting and the others aren't. But the good distinguishing feature here is the yellow gills, and of course the lack of a veil or any sort of basal structure on the.
I just think that they change color from yellow to brown as they age, and then when it gets colder, it happens too.
Well,
I've seen half of these this year, and that's what it seems like.
Well, the place where I was yesterday is private property, and the owner of the property looks for these as well as Tricholoma portentosum to collect and eat. And he was out a few days ago and didn't see any. And there were a few out yesterday, and they looked pretty fresh. And they looked like this. They had brown caps. I mean, it might be because if it gets really cold. I mean, there's been some cold nights here.
Yeah, we've been getting the cold.
Yeah, and sometimes they're yellow, sometimes they're greenish. The Polish name for this, Ganska Zielonka, I believe, means green goose. So sometimes they're green on the cap. So as Igor said, it's a species complex. But in this same area where I found ones that looked like this yesterday, brown, sometimes they're yellow on the cap.
So are all of these yellow gilled ones, or do they all get thrown into that same?
Pretty much. I mean, there's also Tricholoma subluteum, which has gills that are tinged yellowish. Tricholoma portentosum has gills that are tinged yellowish. Tricholoma sajunctum has gills that are often tinged yellowish, at least parts of the gills. I think maybe toward the margins.
But it's
Um,
important to realize that all these have white gills that are just tinged with some yellow, either on the margins or
Yes.
closer to the stem. Whereas equestre, they have bright yellow gills from the get-go.
Yeah. Yeah. Completely yellow. Yes, exactly. Yeah. Completely
Mm.
yellow like these. Hmm.
Yeah, it's one of my big lessons for this year is I know nothing about tricholomas.
Yeah, they're tough.
Can you zoom in, Chris?
Yeah, what do you
It's just want to see a closer view of the gills and the attachment.
Yeah, even the gills are faded. They're sort of dingy yellowish brown. But the young one has brighter yellow gills.
Mhm.
That's the natural color. But as they get older, they fade away into this.
Yeah, but this one's also brown on the cap. Look at the margin.
Yeah.
Yeah. Well, I mean, I've just seen some yellow ones in the Pine Barrens when I was out on Sunday with Nina and John.
This is also probably a spot that gets a fair bit of sun. I don't know if that has anything to do with it.
I think it's the cold weather. Honestly, the spot I was in yesterday, I found one of these that had a yellow cap, and that was the one that was old and had obviously been there for better part of a week. When I tried to harvest it, it just fell apart. And the brown ones were in very good condition. And as I said, the guy who owns the property, it's only about 2 or 3 acres, and he goes out there from time to time and he collects these to eat. And he didn't see any a few days ago, but they were out yesterday, and they looked like these. They had the brown caps, and I was scratching my head over it because I had to sort of review it myself to convince myself that that Tricholoma equestre can have a brown cap.
Yeah, I was just, snapped this one photo, and then when I was looking back on it, I took the AI suggestion, and I just couldn't find anything better.
Oh,
Yeah,
that's a good photo. It's a good photo. Very good.
There's one Tricholoma. So this Tricholoma is from section Tricholoma, but there's another second biggest section in Tricholoma called Genuina, and the species name is Brunoluteum. So that one has yellow gills and a brown cap, but the brown is more kind of reddish brown, and it's
Yeah, it's
smaller.
kind of a shinier cap surface also.
Yes. I mean, Genuina is a very easy to separate from the ones from section Tricholoma. They just have a different look to them, the entire thing.
Yeah,
But
there's a
again,
different gestalt to those.
different gestalt. Yeah.
Yeah.
But I'm just saying that there's other Tricholomas with yellow gills out there, and not necessarily from section Tricholoma.
Yeah, as you said though, Igor, most of them are the gills are basically white with yellow tinges. Although I'm not sure. What about intermedium? Does that have yellow gills?
I don't have a concept for that species. I think it's
Yeah, I forget.
I think it's like
One thing that separates equestre from the others is equestre occurs late. You're not going to probably see it even in New Jersey until mid-October or November. We don't see it here until late October.
Do you have anything else you want to show, Chris?
No, that was it.
Okay. All right. I'll invite others if they have something to put it in the chat. We can move on. Brandon, you were the first to email in, and I know you have some of these. So are you ready to go? I'll start showing them.
Yeah, I can go.
Okay, great. Let me
Yeah, I think are my first two Tricholoma.
yeah.
Oh, must be the season.
Oh, it is the season. That's when they come out.
Okay.
Yeah, so I was calling this
Are
one.
you seeing it before I go to part? Can you see my screen?
Yeah.
Yes.
Okay, good. All right.
Yeah, so I call this equestre as well. Although the gestalt is a little bit different than than I'm used to
Ooh.
seeing. And the cap is, got tinges of reddish orange on the yellow background. And this is grown in the pine barrens. I mean, sand was just covering everything I found this day.
So you see this one is like in the middle. We have the yellow ones. We got the ones from Chris that are brown, and this is like kind of in between. It has both colors. It has the faded yellow and the reddish brown sort of in there too, mixed in. One thing I wanted to say before I forget, if we really want to find out about these equestrian complex, we should look into the Michael Quebec publication, which is 300 pages long, and I'm pretty sure they say something about these things in there.
Yeah, I have been putting off tucking into that because I found a bunch of Tricholoma this day that I was down in the Pine Barrens. But yeah, just a equestrian group or complex.
Yeah, I agree. Look at the gills. Close gills, notched attachment, yellow, white stock, more or less white stock.
Yeah, a little bit of reddish brown on this stalk, but
Yeah, a little bit there. Under
subtle.
pine,
Yeah.
late occurring.
Nice photo. That one looks at least clean on the underside.
Yeah, the ones I've seen when I've been in the Pine Barrens are like, I wouldn't attempt to take them home to prepare them as food because they look like this.
Yeah, I mean, even
and by the way, it should be mentioned, ironically enough, because I'm discussing this species as a presumed good edible, there are fatalities have been recorded in France and Poland about 15, 20 years ago associated with this species. Some blood disease or something. But the people who suffered these maladies apparently consumed approximately a quarter pound of mushrooms each day for like 5 days in a row. And it's also believed that they may not have been cooked very well and may have even been consumed raw. And so the suggestion is that Tricholoma equestre actually contains a potentially lethal toxin, but most of it cooks out or passes through a person's body harmlessly and is just expelled. But there's a tipping point with these mushrooms. That's what the evidence seems to suggest. I still eat these when I find them. I don't eat them more than one day in a row. They're very good edible in my opinion, but this needs to be said about this species.
Thanks, Dave.
Is this considered to have a notched attachment, you say?
Yeah, pretty much all Tricholomas have a notched attachment. So the gills, they approach the stalk, and then they sort of disappear right before the stalk. But if you look very closely, and you might need a little magnification, like a loop, you'll see that the gills are attached, and it's there's a little notch, and then the attached part is not particularly thin, but the actual attachment comes as a result of after this notch.
Well, I can see, yeah, particularly on the lower part
Yeah,
there.
there's
kind of a broad notch, I guess.
Yeah, it's a broad
I've
notch.
seen some where it's more of just a slender little dip just before it gets to but this one's, I guess, but that's true. Is that true that's a notched attachment is typical of all species of Tricholoma?
As far as I know, I don't know about the ones with the annulate ones, the ones with like the Matsutakis, because I don't find those. The only ones I've ever found were in the Pine Barrens. I didn't study them that long because they ended up being eaten before long. But yes, and maybe with the younger fruit bodies, the whole notched sort of thing occurs in a tighter space.
Right.
But yes, my understanding is at least all these exannulate Tricholoma species have the notched gills.
Thanks.
All right, and then Tricholoma.
Can you tell me now how I'm able to pull up my pictures that I sent to myself while I'm online here?
Okay, so what you're gonna have
Peter,
to do is
can-
ask it to
can
allow
you go
to
on
share
mute?
your desktop. And she'll allow you to share.
All right, well, he's working that out. I hope I didn't mute somebody else on the call here. Okay. Sorry about that, Brandon. Go ahead.
No, it's fine. Yeah, so this is another Tricholoma, I believe. And like Chris said, these things I find them difficult. But you can see this one this is a longer stipe, thicker stipe, to a bit more of a, not conical per se cap, but it's got a bit of a more of a bell shape to it from the other angle. Maybe you can see it better. Yeah, there you go. Kind of not broadly umbonate, but kind of rounded. If you can zoom, I think you might be able to see there's like a bloom or something going on. Maybe some darker streaks, radial streaks. But nothing too dramatic in terms of action on the cap there.
Yeah, radial streaking is pretty common in genus Tricholoma.
And that's I was thinking something like subjunctum.
No, I
No,
don't
it's not
think
that.
this is that.
This is fumosoluteum.
What is it? Fumosoluteum.
Mm-hmm.
So that one is one of the ones that is supposed to have like a smoky thing going on the cap based on fumosa.
Yeah, fumosoluteum, smoky yellow. Yep.
Igor, this is the one with the yellow gills, right? The fumosoluteum.
No, has white
Oh,
gills.
I'm sorry. I'm what's the name of that one? I forgot. I'm sorry.
Which one?
The one with the yellow gills.
Equestre?
No, the one that looks like fulva, but it has yellow gills.
Oh, Brunio-luteum.
Brunio. Okay, the luteum threw me off. Sorry.
Yeah. This one is definitely section Tricholoma. It's not genuina.
All right.
And
Thanks.
I think I came across the name fumosoluteum before, and was that one of the ones that that kind of stains reddish or pinkish when bruised, which I think maybe you can see here.
Yeah, I think there's some discoloration of the stipe. Yeah, I think it turns more, I don't know, kind of a yellow brown or something, or brownish or reddish brown. I
Okay.
forget. I mean, when I see the cap, I know what they are, but I don't really pay attention to the rest of the morphology. I shoot more, but there's just no time at 4As. You just give it a look,
Yeah.
give it a name, and then move on.
And what is that giveaway on the cap for this? Is that kind of not not quite brown radial streaks or dark radial streaks, but kind of more subtle ornamentation on the cap?
No, it's just the color. It's this warm yellow with just dusky with some. Yeah.
Okay, cool.
Yeah, I think that's what it is. It's just faded, and maybe frostbitten or something like that.
Yeah, I think in that last photo of the cap, you can kind of see where it would get that name, right? Kind of smoky yellow. Makes sense. All right, cool. Thank
Yeah,
you.
the subjunctum has these darker fibrils, radial fibrils. They're black over a yellow background or something like that. So it looks quite different. It's still section Tricholoma though.
All right, let's move on to a non-tricholoma. This, I don't know. I feel okay about the name. What is it? Yeah, right.
Pisolithus.
What is it? Meridionale.
Isn't that Pisolithus arises? Did you cut them?
I did, but the polyrhizis, I've never seen it present this way before. The way it has
Yeah.
these super thick rhizomorph bundles, as the description for this species describes.
Yeah, you're right. This is Scleroderma. Pisolithus looks obviously very, very different on the inside. I was just misled by that stipe, kind of a fistula looking thing.
Yeah. Yep.
That's a weird one.
Yeah.
Yeah, I've
Very
never
much.
seen this one before.
Because
Yeah,
I
it's
was
pretty
looking.
stipitate,
Ye-
capitate thing.
Yeah, but if you can zoom in on that stipe, maybe on the one on the left, you can make it out more. They're actually these like tendrils, like tightly packed tendrils. I guess that's the best. I think it was described as rhizomorphic bundles on the species description for this one.
I know, it looks like a solid stipe to me that sort of crumbles away at the bottom or becomes channeled or hollowed out. I don't know. Maybe I'm wrong.
Yeah, you are.
But you can see the stipe. You see the cap where the galeba is, where the spores, and then you see the stipitate structure below it, and then it goes into this root or a bulb, and it becomes this mess.
Yeah,
I've never seen sclerodermas that look like this. I mean, this is a mutant of some kind.
it really is a mutant. It threw me for a while.
Wow.
If we
Did you save some? This would be an interesting one to have
Yeah.
sequenced. Yeah, good.
If we can cycle through the photos, are there more photos?
Yeah, good.
Maybe of
Wow, that's a stipe.
yeah, so that's with the bundles removed. So that those tendrils underneath of this very obvious stipe, that's like the stipe part, and then you can see the remnants of those little tubers coming out of it.
Yeah, like
Interesting.
dendrites or something. Yeah. Ugh.
Scleroderma verrucosum seems to, I'm seeing a lot of pictures of it. I think that might be what you have. It's pretty much a stalked scleroderma.
Can you put that in the chat, Hunter?
Yeah.
Okay, I think that was the last picture there. Okay.
Was any of that like
check.
mass, like was that all under the ground?
Yeah. Yep. There's an in situ photo somewhere, but that verrucosum doesn't have these tendrils,
Okay.
right?
Yeah, I agree. I think this is meridian. This looks more like meridionale than varicosum because it doesn't have those rhizomorph bundles.
Yeah, and they're tough to take photos of. Maybe zoom in on this.
I'm seeing pictures of what looks like those similar bundles, but that's just pictures. So which was the one that you said, Jason?
Meridionale.
Okay. Can you put that in the chat? Thank you. That's
Yeah.
really
Pretty much the research that I did on this one, I was looking at septendrionale, because that's the one I'm familiar with that has I was looking at polyrhiza, the other mosaic one, and then septendriale. But septendriale has whitish rhizomorphic bundles, and these very much are yellowish, fitting with the kind of the rest of the mushroom as well.
okay. Yeah, I'm seeing that without.
Yeah, that yellow color is diagnostic too. I've seen this with John and Nina at Franklin Parker Preserve.
Oh, cool! You have, because I'd never seen anything like it. Definitely looks a little bit alien.
Yeah.
Cool.
I would say the underground structure reminds me most of like a calostoma type.
Yes.
If that
Same.
makes
Yes.
any sense.
Yeah,
So-
totally reminded me of calostoma, but the black, purple inside was like, no way.
and they're big. You saw it next to that tape measure. They're like, that whole structure is like 10 centimeters.
Yeah.
Which calostoma can get big too.
Yeah. Very interesting.
Yeah,
Okay.
big cool find.
Nice, thank you.
These, I don't know, scales or whatever did make me think initially for a second that it could be one of those Pacilithus things too. I think those are on there. But this is really interesting. All right. Tarzetta. Largest cup. All right.
Oh yeah, these are cool. I think I have the genus correct, and I'm pretty sure. What's the species I put in there? Cupularis.
Yes.
I think that's probably what it is. Just want to check a few more resources to kind of confirm it because the spore measurements that I got were big. But these things are tiny, less than a centimeter and a half, I think. You can see they're kind of granular on the outside and have like a toothy margin, torn toothy edge of the cup there. Yeah. There probably is a foot hidden underneath this sand, but again, sand makes things difficult to ID things in the Pine Barrens sometimes.
Oh, those are so pretty.
Yeah, they're cool.
I'm sorry, what was the genus again? Was that Humarium?
I'm calling it Tarzetta.
Oh, okay. Yeah, I guess Humaria is bigger.
I don't know. I think there probably are some small ones like this. Are they granular on the outside though?
I'm not sure. I'll check.
Okay, yeah. And then the spores. Did we do cycle through to the spores yet? Okay, there we go. Sorry. They're just a little bit bigger than fit the description for Cupularis, but I mean not bigger by a couple of microns. So that's probably what it is. That last photo, the small one up top, I included that because this is the best one, even though it's out of focus and probably on a different plane than all the other ones. That shows that it's bigutulate. It's got one of those oil droplets at either apex there, which also matches for Cupularis.
You could sort of see them in the bigger ones too.
Yeah, the thing with the bigger ones that also gave me pause is it looks like there's more there. It's bigutulate at either pole, but then there's like little ones in there too.
Yeah, something going on inside. Yeah.
So Humaria has ellipsoid spores that are bigutulate, but the exterior of the cup, and there's only one species described on Myco-Quebec, is the exterior is hairy and not granular. So yeah. Oh, and there's an ascus. Let's
Yeah,
see
a big one. That was what shocked me on this because this is at 100x. So that's what 10, 20, 30, 300 microns. Pretty big.
Yeah, these are big.
And if you zoom in on the foot there, there's it's got like a little bifoot or something, some action going on.
Yeah, it's as much I can zoom in.
Yeah.
Yeah. Yeah, nice photo.
Thank you. Yeah, I think I'm pretty sure it's Tarzetta cupularis. I mean, based on the images that I come across for that, it looks exactly like that thing.
Brandon, how large were the spores?
I definitely have the measurement. I have the measurements in there. I want to say they were like 23-ish.
Okay. Yeah, I see Humaria spores 22 to 27 by 10 to 15. But the exterior is hairy, not granular. Otherwise, there are some similarities.
Well, are you familiar with Tarzetta cupularis?
No.
Look that one up real quick.
Yeah, let me check that out now. I'm right here on Michael Quebec. Oops.
Yeah, I would call that granular.
So, Brandon, did you rule out Tarzetta catinus?
What is it? Catinus?
Yeah, CAT.
Catinus. No, I have not.
Maritel has an observation of that from New Jersey, and it looks like it has slightly larger spores.
Oh, cool. Nice. And looks pretty similar.
Yeah, and she's got a really nice observation that has those long asci.
Cool. Yeah, that's definitely one to check out. Catinus, Catinus, right?
Yeah.
Yeah, it's on Michael Quebec. I was just looking at it.
This one of your burned areas?
No. At least as far as I know. No. Is that one maybe associated with burns? Because there are some of these Tarzetta and cup fungi that are burn associates.
Yeah, the edge on yours, like you said, that toothy edge, the rosessa, I think that's how you say it.
Yeah, I'm looking on Michael Quebec. And that's one of the features of Catinus is the toothy margin in age, after it's mature.
I think that's a feature of both Tarzetta catinus and Tarzetta cupularis.
Yeah, it's definitely and I was just going over Catinus, which I'll definitely compare there. It's definitely a feature for Cupularis also.
Yeah, it's one of those two, very likely. It's Humaria. I mean, I was just playing devil's advocate there. But
Yeah,
it's not.
but
that has a different exterior.
yeah, but that it's good to compare and know that if I ever find something that's hairy, look at Humaria, right?
Right.
But yeah, Jason, this I think Catinus looks pretty good. But I'll compare. But yeah, probably one of those two. So thanks, guys.
Pretty good. I think you have another one. Yeah.
Yeah, it's a Galerina. Or it's an LBM, right? Uh, found at the edge of an Atlantic white cedar swamp. Um, not necessarily in the moss.
I hope you're doing well, period. I will let them know.
I found it in like a little crevice under a tree root. Kind of boring looking little thing. It does have the marginal striations. I'm calling it a Galerina. I don't think it's Hypnorum. It doesn't look like Hypnorum to me. Maybe it's Pallidosa, but I'm not super familiar
No,
with that
not
one.
Pallidosa has Pallidosa just looks different. It has a lot of velar material on the margin
Yeah,
and velar material on the stipe.
There
And
is
it's
velar
usually in
material.
moss. Was this in moss?
It was in moss and like a little hole. Yeah.
So, but it was on wood. You said.
No, it was under like it was in a little hole under
Oh,
a root. A root.
did you get spores for this?
I did. But what I was
Yeah,
going to
I'm
say,
wondering if it might be tubaria.
there's velar remnants on the stipe anyway, is what I was going to say.
Oh,
Something right there.
a little bit, yeah. I don't think this is Pallidosa though. Oh, spores. Okay, let's see.
It's a little paramecium kind of shaped.
Yeah, I don't think
Oh,
those
it's got
are
a
tubaria spores. At least not Furfuracea or the similar ones. Okay, so I withdraw my tubaria. Oh, actually, no, that one there with the gutules. That does not look unlike tubaria spore. Did you get a spore print color?
yeah, it was brown.
Yeah. Well, so the Galerinas have rusty brown usually. I mean, not dark brown, but rusty brown. And the tubaria tends to be more like light brown, like yellowish brown. I mean, it's pretty close though. So I'm not sure how good a character that is to fall back on. So that's a hymeneal cystidium there. Well, that's really there's a species of Galerina called Tibia cystidia, something like that.
I think he's gone on mute. Brandon, we can't hear you.
Oh,
Yeah. Galerina to
Tibia cystidia, something like that. It has cystidia that look like that. Tibiiform, they call it, where it's just like a straight cylindrical stalk and a round head.
Mhm.
There might be more than one species of Galerina that can have cystidia like that.
Yeah.
But I think Galerina makes more sense than tubaria. So
Okay, cool.
Good. Okay. Peter, do you have something that you'd like to share from your desktop? You're on mute. Calling Peter.
Okay, let's see.
All right, I'll stop share and you can go ahead.
Okay, not sure what's going on my computer right now. I just wanted to share a Auricula polytricha, and I'm not sure if I'm being heard or not. So
We can hear you,
okay,
but you aren't sharing anything yet.
right, okay. So I'll share. I'll try to share, and I'm not sure how to do that with my computer. So
You
I will,
started. Okay.
let me see if I can do this.
We see your desktop with the guy skiing. All right.
Yeah, right. So we'll get into all the personal stuff in a second, I guess. So I'm just not sure how to go into my email to show you that. Let me try this. And so here we go. And all right, you see that?
Okay, yeah, bring it
All
up.
right, great. So this is in Englewood, New Jersey, a couple days ago. I'm like, all right, look at that. Nice fresh specimens of a wood ear. And I have them now on my table ready to cook. And I was wondering if anybody else was going to confirm that it was Auricula polytricha, because there's a couple species out there. And I wanted to confirm with you experts that it was polytricha. It doesn't really have any hairs, so to speak, but if you look really close with a hand lens, then you may see that it does have hairs. And
Yeah,
otherwise,
they're quite short, but it's definitely has that fuzzy exterior.
yeah, definitely fuzzy.
Yeah,
Yeah.
fuzzy or just a sort of a matte,
Mate.
scruffy.
Yeah.
I think all of the auricularia have that feature.
Uh-huh. And you call it Auricularia as opposed to Auricula?
Auricularia is the genus. I think. No, let me double check that. And yeah, Auricularia is the genus. There's a species named Auricula.
Right.
But I don't think that one occurs in North America.
Okay.
I think that's the one that's basically sold dried in Asian markets, imported.
Yeah.
We have a few species in North America. And first of all, if you know whether it's a conifer or hardwood, that's a starting point.
Good question. Well, very hard to determine because it was a dead stump. And
Yeah.
did have some bark on it, but I really wasn't paying attention to it at the time. But I'll revisit it in a couple of days tomorrow and see what kind of stump it is by its bark.
Now, what was the
Looked like a
species that
hardwood
you
stump.
suggested? Auricularia. What was the name of the species?
Polytricha?
Polytricha. That's one that I think I've seen come up on the websites lately. So, so that's a name that's being used, I believe, in North America. Let me just look and see what I can find on it.
Can you go back to that first photo?
Okay. Good luck with me. Let's see if I can do that.
Scroll up, maybe. There you
Thank
go.
you. Interesting.
Try clicking outside the photo.
How's that? Better? Little
There
faint.
it is.
There we
Wow.
go.
There it is.
Magic. Unbelievable.
Yeah, that looks like some hardwood.
It seems like, yeah, could be oak. And
So I think I mean I would need to check this out more. But now it looks like to me like maybe auricularia, auricula.
yeah, that's another one. Uh-huh.
But I think that one might be European because that's probably where the it was one of the first species was named. It looks like the Asian one is Auricularia cornea, and I think it sounds like, just based on my search I just did, that polytricha is has now been changed to this name.
Oh boy.
So, but in North America, we have several species. On hardwood, one of the more common ones is Auricularia angiospermarum. angiospermarum, which basically means fruits on hardwood trees. Hardwood plants.
Angiosperms.
Yeah, and then on the conifers, usually pine, there's Auricularia americana. So my guess is these would be Auricularia angiospermarum, spermarum, hard to say.
Yeah.
But there are other possibilities, and I'm not sure about this polytricha. Is that the one that's sold in the Asian markets? Do you
That's
know?
what when I did a little bit of research that I did in the books that I have, and none of them had a really good photograph like this. They had just like one or two fruiting bodies, but I thought that this was really very clean looking photograph.
Right.
But no ruler in there, or no penny or dime or nickel or ruler. But I did say about Chinese people, this is the one.
Oh, so this is polytricha is the one from Asia. So that we probably don't get that one here. Unless if it has, oh wait, I know, I remember now. We had this discussion before. Is Dorothy, are you out there? I guess Dorothy's not out there. Dorothy has found polytricha on her property. Dorothy Smallman. I'm pretty sure we've had this discussion. And it seems to be a species that may have just been imported from China. But apparently it is here. So, and I think that Dorothy was finding her tree ears on like an oak or something like that in her yard. So, so that would seem like a reasonable proposal here. I would also check out Auricularia angiospermarum. But I don't even know how to tell these things apart, and you might not even be able to without sequencing, because they all look pretty much alike. There might be like difference in spore size or something like that.
There's a species of this that is commonly found on Tree of Heaven, which is what this bark might be. Tree of Heaven is native to East Asia.
Yeah, I got plenty of those around here, those trees, of course, but not in this vicinity because it's a nature preserve, Flat Rock Brook Nature Center in Englewood, and I think they don't have any because I think they cut them down, which is a good thing. Yeah.
Isn't there a whole process of getting rid of those things? Like not just cutting them
Yes,
down?
correct.
Not to derail the convo, but
right. Yeah. You have to poison the stump, basically.
Yeah.
Yeah. Nasty
right,
plant.
well, if this is Ailanthus and it was killed with some sort of herbicides, I'm not sure I'd be eating these auricularias.
Well, the stump is 1.5 feet tall, and they probably wouldn't have used an herbicide on there as to kill it because it's a nature preserve. I can find out, but a good question what the rules are there.
I manage nature preserves and I use herbicide on Ailanthus. I cut the stumps and then I dab the herbicide on the cut portion of the stump to prevent resprouting.
In the middle of the night?
No.
Did you go out there with a crossbow also and take care of the deer?
No.
I do have people with crossbows shooting deer, but it's a separate issue.
Yeah. You're the fellow who works for the parks.
DEP, right?
No, yeah, it's separate preserves, not this one.
Yeah,
but
right. I mentioned, yeah, great. Yeah, we did a walk around our fenced in area. We 4 bucks that were just huge, crazy. It's not good here in Northern New Jersey.
Yeah.
Not to get off the subject, sorry.
All right.
Okay, well, that's my addition, my portion. Hopefully it's helpful. Thank you, everybody.
Thanks for sharing. You can stop that share now, if you would.
Okay, yes, let's see. Haha. Let's see, I'll remove that. And I'll delete this and delete.
Yeah.
Any better now?
No, you're still sharing. There should be some stop share red button either at the top or the bottom of your screen, your Zoom window.
Okay. I'll probably just sign out is what's going to happen.
All right.
All right.
Join back
Sorry
in.
for my unintelligence relative to these computers and sharing stuff.
You're learning. We're all learning. You're getting better. Don't worry about it.
Okay.
All right.
Good night.
Good night. Did that end it? No.
It will in a second.
Okay. All right. There you go. All right. Does anyone else online have something they'd like to share from their laptop? If not, I will go to the email. Okay, going to the email. Hunter, you are
Okay.
up.
I can go, or if anyone else has anything they want to show first, I'm fine with that. Okay, so yeah, it's getting pretty sad around here mushroom wise. It's just wintry and dry. But I found this at the base of a ash tree, I think, or it might have been an elm along a bike trail, and I just noticed that yellow line, and I thought that was interesting, and I've seen that on another mushroom before that I thought it might be the same kind, but and everyone probably knows exactly what this is, but I'm not 100% clear of what this is. It was solitary, and I'm sure someone knows what it is.
Looks like Armillaria gallica.
Okay, thank you. That's it. I didn't want to say it, but that's what I was thinking. It's just a honey mushroom. Okay, so what's the yellow line?
I don't know what the yellow line is, but that yellowing is very common with gallica.
Okay.
Yeah, what gallica has is yellow stipe base, and it's also bulbous, and they grow in the ground as opposed to directly like on the wood, like on a stump or something.
Yeah, and solitary too.
Yeah,
Generally?
they're solitary or just gregarious, but never like in big clusters like
Malay.
Melia.
Yeah.
Yeah,
It,
yeah,
it,
they tend to be on the smaller side.
That's
like
what I
this.
thought. I was thinking it was a honey mushroom, but I don't know. Just wanted to.
that yellow in like a ring zone is pretty interesting, though.
Mhm.
It's not like something you often see in gallica at the very least.
Yeah, I have this picture of. I think I've shown it to you guys before. It's a huge mushroom, with a very similar yellow ring on it. It probably is the same thing, and it probably is Armillaria gallica. But yeah, maybe it's a drip line or something, or I don't know. It's interesting.
It's hard to tell. We got to read that. Probably the description says something about the nature of that yellow ring if it presents all the time. But the partial veil is above that. You can see that, right?
Right. Yeah.
Yeah.
Right.
So I don't think it has to do anything with the ring itself, since it's kind of below and distant.
Right.
Right. And I was just noting that it's interesting that it looks like an annular zone, though, right? It really does from like,
Yeah, like
if
a ring zone or something like that. Yeah, but it's not that.
Well, gallica has a partial veil that's somewhat coordinate, and it falls apart pretty quickly. But I think I'd have to review this. I think it might sometimes have a yellow margin at first, or there is an Armillaria, I believe, that has a yellow margin on the partial veil. And if the partial veil collapsed onto the stalk and disintegrated and left part of it as a ring, as we see on the uppers part of the stipe here, Perhaps the margin having collapsed a little bit lower down may have
Right.
that yellow. But I think we want to find the Armillaria species that have the partial veils that are partly yellow, or especially along the margin.
Yeah.
So,
I've seen that. I might even have a photo like that, where, it's white by the stipe, but if you go out a ways, there's a yellow zone on that partial veil. So it could be that this is all partial veil that's just collapsed down.
that's what I think. And
Yeah.
you just have to find the one. Does Gallica have the yellow margin? I'm not sure. I'd have to review that. Let me look.
Yeah, I don't, I think it might be, but
I
I'm
don't
just
see
don't
it in
remember.
any description.
No, okay.
But I mean, I only read a couple, but no.
Okay. There's definitely something that has that in the honey mushroom family, if you will. All right,
Well,
well,
thanks.
nice picture.
It's okay. Don't feel very well, whatever. So there's a few things here, I think. Yeah, there's the black and then the hairy stuff, and then it looks like a white crust below that. But I took the picture for the middle, and I was wondering, and so I've had no time. I've been really, really busy to do any looking into it. But Lila, you and I in New Jersey saw something fresh that I don't know if this could possibly be it later. That you took such a beautiful picture of, but it was like more white and sort more like maybe leathery in appearance, but it had similar structure. Do you remember that? It was
no, but
okay.
I'll look for it.
Yeah, it was a really pretty picture. But does anyone know what this is?
Is that the tomentose growth mode of one of those Coprinellus?
Oh,
Coprinellus. Coprinellus. There's a Coprinellus that has domestic. Is it domestic? Blah blah. The one that starts with domestic. I think has like mycelium that looks like this that surrounds the base of the stalk.
Right. Yeah. That is.
I think that's Coprinellus.
Coprinellus. That was my first thought.
That's an interesting idea because it might just be the myceliums there and not the mushroom.
Domesticus or
I think that's it. Yeah, I think that's the one. Domesticus. Let me look it up. By the way, I put something in the chat about an Armillaria that might fit better than Gallica.
Thanks.
Yeah, I was about to put one in there too.
What are we going to put in?
I was going to put in calcescens. Did you look
Yeah,
at
I'm
that?
putting in
wondering.
sinapina.
Yeah, I'll throw calcescens in there as well.
Well, sinapina has a mustard yellow partial veil, and a few of the photos on Michael Quebec show like a ring zone below a ring.
Yeah,
okay.
And the complex annular zone was something that caught my eye with calcescens.
Yeah. Okay.
And also mentions yellow. So
there you go. That's another possibility.
I am seeing this domesticus pictures of it growing out of stuff that looks kind of like this, but maybe more orange. That's very interesting. That could be it.
I kind of missed the beginning of when you presented this, and I heard you say, "Does anyone have any idea what this is?" and that was the first thing that came to my mind. Is this? Did you touch it?
I did not. I don't think so. No, I don't think so. And this is in southern Wisconsin, like last week, and um, mixed woods. There's it's some white pine, some oak, some juniper. But this was dead, whatever it was growing on. I don't even know.
Yeah, because I think that Coprinellus that I was talking about, I think that might be wrong. Because this looks a little bit more like kind of complex, like rhizomorphs with some tendrils coming off of rhizomorphs.
Mm-hmm.
How big is this patch that we're looking at? A couple inches?
Yeah, for sure. It was decent size. I mean, probably at least 6 inches by 6 by 6 or 8 by 6 or something. It was quite substantial.
So this part from here to here is like 6 or 8 inches?
Yeah,
Yeah, okay, that is big.
yeah, yeah. And it was on the underside of a log. So Lilia, are you able to look up that other thing?
Yeah, that other thing I found was white, right? I think
Yeah.
it was young, like Copelandii or something. I don't think it was much more thick and
Yeah, leathery kind of.
yeah, much more substantial than these are quite wispy, I would say.
Yeah, I just thought maybe like in an advanced stage they
Mm.
would turn this way, but I don't know. I guess you don't.
Yeah.
Yeah.
I mean, what is it? Chrysorhiza? Uh, what is that? Is it a Hydnophlebia? Chrysorhiza? Has those orange rhizomorphs. They're normally thicker than this, but I guess that's something to consider there.
Okay. Yeah, I'm looking at that. Yeah.
Sorry, why did we nix the Coprinellus?
I didn't.
Oh,
But
okay.
yeah, no, I think I think Brandon was saying this seemed to have more structure, right? More complex structure, but
Well, that and the size, you know. Although I'm not that familiar with that other Coprinellus either. So
yeah. Me
Well,
neither.
was this buried at all, or you flipped this thing over? Or...
No, it was exposed on the underside, but not buried at all. Yeah. Okay.
Yeah, I see two species of Coprinellus that do that.
Oh,
Domesticus and then radians. I'm trying to figure out the difference between the two.
yeah. And as I scroll through just pictures of Domesticus, I am seeing some where these growths are less deep orange and more like mine, more kind of washed out looking. That's neat. So if it, let's say it was Coprinellus, what's going on with this? It's mycelium. This is carpet-like growth of mycelium. Called an ozonium. Yeah, interesting. I could go with that. I wonder if that if the white crust fungus is a like pre-stage or that's a separate crust. And then what about the black stuff above? There's sort of a lot going on here.
There is a lot going on.
Yeah.
Yeah. Jason, do you have something on your screen you want to share that illuminates on this? I'll stop and you can share it if you do.
I'm just looking at photos on my naturalist of the two, and I was just reading
Okay.
Mushroom Expert, which I think is what Hunter is reading from, the ozonium. Looks like spores separate radians from domesticus, but we don't have fruiting bodies.
Yeah, we don't even have a fruiting body.
Yeah.
Right. It's very interesting.
Funny, I brought up the wiki for that domesticus, and it reads the fire rug ink cap gets its name from the bright orange carpet of hyphae grown around the mushroom. The covering is known as an ozonium, which resembles an above-ground mycelium.
Wow.
The ozonium is not always present and can also grow in the absence of any mushrooms.
Mm,
All right, there you go.
that sounds like it.
Oh, okay.
Yeah, because I only brought that up because I didn't know that it could grow in the absence of a mushroom. I kind of figured that the mushroom fell off of this thing. And it's one of the only coprid, I called it Coprinopsis. It's a Coprinellus. I get those confused all the time. It's one of the only ones I can ID because it's got this, would you call it a fire rug?
Fire rug.
Yeah, that's cool.
Love it. Oh, well, this is good. Yeah, very cool. Like I think we're on to something. Yeah.
Yeah. And I think we probably see this and gloss over it a lot in the field.
Yes.
Um,
Certain I've seen it before.
yeah, I feel like I've seen it too, but
Yeah.
figured there was no way to identify it and kept moving, probably.
Yeah.
Thank you for bringing it to our attention.
Oh yeah, my pleasure. Mm.
All right, and good help from Jason and Brandon on it. Thank you.
Yeah, thanks guys. And so I was wondering, I know Brandon, you showed this white fungus on juniper a while back, I think. And
Yeah.
is it? Or
Did this thing have pores?
I my pictures are terrible and I can't even say with certainty, but I think so.
It definitely looks like Dendrothele. What is it? Nivosa? Dendrothele
Nivosa.
nivosa. Is that a juniper? Is that a
Yeah.
yeah?
Yeah.
Doesn't look like our junipers, or at least the ones that I see in New Jersey.
Yeah.
Looks big.
It's not too big. I can put a picture in the chat of like a standing one from where I was.
Looks like you're on a cliff edge there.
Yeah, I am. It's actually a really beautiful spot. I will do that. Hold on.
Maybe the smallness of everything in the background. I didn't realize that made this thing look big.
Yeah.
Yeah, that's probably it. Yeah.
Oh, but yeah, it's probably Dendrothele nivosa if it's on a juniper.
Okay. Yeah, that's kind of what grows there, I guess. Yeah. And so and that's a parasite. Is that right?
That I'm not sure of.
Okay.
Yeah, I'm not like super familiar with the actual fungus.
Okay.
But my experience with it is, I mean, I see it on like 50% of the junipers that I see in the forest.
Yeah.
And they seem to be doing fine. So I don't know. I think it's something that maybe just infects the bark, but that's a total guess.
Yeah,
and doesn't work its way into the kind of the heartwood.
right. Can you put it in the chat? The name? Thank you.
Can you folks see that beautiful shot of the valley below?
Oh yeah, that's great.
Yeah.
Yeah, there's a section of the Ice Age Trail called I think they called it Gibraltar Rock or Rock of Gibraltar. I'd never done it before. It was a real sweet spot. Yeah. Really.
Cool. And the fungus was on this tree over
You
here,
know, I don't know if it was that one, but
something like that.
yeah,
Yeah.
yeah, that was
Okay.
about the size. I don't think it was that one. That one seems more weathered.
Mhm.
But yeah, but no, I'm pretty positive it was juniper because I've been looking for that fungus because I know one of I think I've told you guys this before, but the woman who does the golden oyster studies in Wisconsin, I can't Ashwara, or I think her name is,
Mhm.
her good friend is studies that white fungus, I think, on juniper. So I've been wanting to find it, and then Brandon mentioned it and found it, and then so
Nice.
yeah. All right, and then I think that's it, right? Or maybe I have one. Oh, I have one more, and
One
yeah,
more.
yeah. This is from like right before Halloween, in northern Illinois, and not really exciting, but I thought I'd show one more photo. Uh, I was thinking clitosopy of some type, but and it had a memorable smell, but I can't remember what it smelled like now. So just kind of wanted to share something, but
You
not.
get a spore print color for that?
I did not even get that, or maybe no, I don't think so. No, I want to say white, but I can't say with certainty.
Yeah, because I'm wondering if this might be one of the terrestrial Philiotas, like mixta or lenta. But those have brown spore prints. Brown spore. Is that a ring on the stalk on that one in the upper part of the little ring zone?
I wondered that as well.
It's as close as I can zoom, but yeah, I had the same question. Yeah.
I think this is one of those Philiotas. One of those terrestrial Philiotas.
Yeah, there's something different about it. You know, it wasn't something I see all the time. Um, Let me look back at my photos and see if I have any more. I don't think so, but let's see. Yeah, it's really hard to tell if it's a ring zone when I blow it up on my phone.
So, Dave, you think this is like a what is it, terrestris type thing?
Well, it could be one of those kind, and it actually looks a little more like one of those kind. But there's also this thing called mixta, and
Mixta.
Lent is more of a uniformly pale cap color, though, so I don't think it's that one. I'm looking up mixta now. I'm on Michael Quebec. Let's see if that looks like it might be promising.
I think the base is
No, mixed brown. Mixta's brown. It's not that.
okay.
Yeah, it's not mixed. That one's a lot darker. Sometimes it's pale, but it's really dark on the disc. So it's not that one. So yeah, one of these things, like, what is it, terrestris? I
I forget
guess.
the name, but that one's pale too, kind of. Terrestris. That something
There's one of these Philiotas. I'm gonna look for it. There's one of
something
these
in
Philiotas
reference
that
to
looks like this.
terrestrial.
Terrestris, yeah, that That's one. It does look like that. Huh.
Yeah, I'm wondering about that ring zone and just the overall appearance. Let me just.
It's interesting.
Oops. Pholiota. So there's Pholiota scamba, and that's a terrestrial one, I think. Let me see what that looks like. There's a lot of these Pholiotas. There's a fair number of them, and those scambas also looks like it's dark. It's not that.
Seems kind of hygrophonous too, right?
Bumosa.
Yeah. A little bit.
A little bit,
Yeah,
at least this one, if it's the same thing.
And I'm
Yeah.
thinking on the one on the left, I think you can get. I can't tell if those are scales or if it's some hygrophonous patterning. You know,
Yeah, I can't tell either.
but you can tell the center of these things is darker, pretty much on all of them.
Yeah.
Yeah, that's what I'm getting here is pretty much all these Foliotas I'm looking at are the caps are darker. Now there is Lenta, but that one's sort of overall pale. Well, actually, no, I'm looking. Yeah, that one looks a little different. Was this so slippery or greasy or sticky or slimy at all on the cap?
I don't think so.
No. So
Mhm.
maybe I'm off base here with my
The
Pholiota idea.
this mycelial fuzz, if you will, up here seems kind of tinged a little bit blue or something, isn't it? Or is that just
Yeah, it's kind of gray blue, but
very blue? Yeah.
yeah. Yeah, it's not great pictures. I'm just wanting to share something and don't have much, I have to say. So yeah, we can move on. But
All right. I think that was, yep, that was your last. Okay.
yeah.
All right. Let me put out a call here. Anybody online that has something? I'll pull up this Quebec calvescens back to the Armillaria that has
Thanks.
the yellow green.
I also put in
Oh,
the
look
chat
at that.
the what is it, Sonapica?
Synapses or something. Synapian.
Sonapica, I think it is. Look at that one also.
Synapina. Yeah.
Synapina, something like that. Yeah, look at that one also.
Yeah. Can you open that
That
one?
one causes yellowish deposits on the stipe.
Look at that little one in the photo where it's just where it's about to break away. There seems to be a whole bunch of yellow.
This one up here too.
Yeah, and that
Yeah.
I think that matches Lila, your kind of theory of how that marginal yellow can kind of deposit down on the stipe there as the thing expands.
Yeah, it just flops backwards and ends up down lower, maybe. Yeah.
Yeah. So
Also,
maybe
there was a lot of like pinkish and reddish tones in your mushroom, Hunter.
yeah.
And I think you see a lot of that in this mushroom as well.
Yes.
Yeah. And the base of the stipe looks pretty similar too with this yellow.
Right. There was some yellow in the base as well. Mm-hmm.
Yeah.
And
Yeah.
it's funny because Igor and I were both like, yeah, it looks like gallica, because I doubt this thing would grow. It grows in New Jersey. Maybe it does and we just overlook them, but I've never seen this thing, I don't think.
I think it's more northernly, a little bit more northernly, so it could be here, you know.
Yeah, yeah.
Check out that other one I put in the
Did
in
you
the
put
chat.
something in the chat here? All right, let me.
Yeah.
Just above the other one. I opened it, and the caps are really yellow too on this one though.
Oh, okay.
Or maybe not. No, wait. No.
Oh, this is sinapina.
Yeah,
Yeah.
sinapina, right.
Yeah,
Oh,
I'm
that's
saying it the wrong
interesting.
way.
Look at that.
Yeah, this is one of those, another one of those that I always question, do we have this one in New Jersey? Like I always come across this name and I'm like, no, it's gallica. But you know, this is definitely a distinct species too.
Yeah, two kind of
Oh.
things going on here.
Wow.
Yeah, look at that.
Gee. Huh.
You know, I outside of Malia, which I can identify usually by the fact that it's has very little ornamentation on the cap, and it's usually one of the earlier ones. I'm not very confident identifying these different species of Armillaria.
Yeah,
Yeah.
Armillaria or Melia, the old Tibessins, whatever, Cespitosa.
Tibet. Well, yeah, Sepistopes, Desarmillaria. Well, that one, yeah, you know, because it grows in clusters. It follows roots of trees, and it has no partial veil at all. And gallica. Sometimes I think I know what gallica is based on the coordinate partial veil.
Well, when gallica looks like when like a textbook gallica, you know it's gallica, but you know, mushrooms aren't always textbook.
No, right. Yeah, exactly. Yeah. So like I say, I most often I end up just proposing genus, identification of genus on Armillaria mushrooms because I just don't feel confident.
Yeah. Yeah. Okay. That's nice to hear because yeah, I mean I was like, yeah, it's a honey mushroom, but which one? Yeah, I was, you know, pretty
Yeah.
positive, but
Even look
They're
at
hard.
even look at these photos. Like the
that's
caps
cool.
on these, they're wet, so they're, you know, they look very slimy and
Yeah.
and bare. But on those last dry photos when they were young, they're pubescent.
Yeah, a lot
Yeah,
of
they have
variability.
the scales that are really just tufts of hairs. Yeah.
Stunning mushroom, really.
Yes.
Yeah, here you can see some of that yellow that's out further on the partial veil. Yeah. Yeah, look at the variability. Wow.
Yeah, that was a cool shot. Just the two of them next to each other.
This one.
Yeah, the two, one of them bald, one of them completely with like pyramidal hairs.
Yeah. Yeah, these are nice.
Beautiful.
All right.
Yeah, it's kind of
Apparently very popular one. They have dozens of them. Yeah, okay, good.
yeah. So what after looking at these, are people leaning towards one over the other, or the calcescens more?
No, no.
No. Yeah.
I
Okay.
Yeah,
don't think it's Gallica though. Honestly,
Yeah.
another thing about Gallica is it usually doesn't form um a, a true ring. Like yours had it was a small ring, but it was a membrane. You
Yeah.
see what I mean there? It's like a
Uh,
membrane.
I don't know about that with Gallica. I think Gallica, not the secondary yellow one, but I think that membranous little
Yep.
nub,
Yeah.
nub, I think that can be I think that's pretty present in Gallica. It might wash
Okay.
away. It might wash away.
Well, what I'm thinking is Gallica usually has a coordinate partial veil, and it's not substantial. It just collapses up and just leaves a zone, or maybe a couple, like some little tufts. I don't know. I have to look at Gallica now. Let's look at Gallica on Michael Quebec. See what they say.
Drop me a link. Just a time check. It's 8:30.
Yeah.
You've got
Yeah, I
some
know.
you want to show too,
Well,
Dave.
obsessing on the Armillaria.
Yeah.
Yeah. I've got a couple of good. I have one in particular that's really good.
All right.
Well, let's move on and see him. Thanks for everyone's help.
Okay. all
Okay,
right.
Michael Quebec has one photo of Gallica. It doesn't
All right.
really show what we want
It's
to
not.
see.
It's not
Okay, so
very.
that's good enough.
Not a
very
good one.
helpful.
All right.
Yeah,
We're
it's
gonna
good
move
enough to
on.
move on.
We'll move on.
All right. So found yesterday in a yard, a lawn under hemlock, and I think this might be the rare Alaskan gold mushroom that has been called Pholiota aureum or aria for a long time, and now it's been put into Cystoderma. So it does remind me of a Cystoderma, but a big, robust Cystoderma with a lot more color
Yeah,
on
wonderful.
everything. So now they're putting this, if I'm
Beautiful.
correct, they're putting it in Cystoderma aureum. Now, this is also, there's another observation of this same collection on INAT, because a guy who was with me also made an observation, and INAT said it was an Amanita, and then somebody else agreed. I don't think this is an Amanita. The gills are too pigmented. This partial veil is very substantial. These are older fruit bodies. They suffered through a couple of cold nights, probably. But this partial veil is very thick. And one of the last photos before we get to the micro shows what I believe can be seen as the partial veil actually extending down the stalk as a sheath. This one right here. This is not my photo. This is a photo taken by a guy who was with me. But look, see how the partial veil kind of transitions into a sheath? And that sheath is kind of grainy looking too. So the texture is kind of grainy, and it looks like it's all part of the veil. So I don't think this is an Amanita. I think this might very well be this Cystoderma. What is
Aureum.
aureum?
Aureum.
Now, so where does this thing grow? Typically in Alaska and the Pacific Northwest, but there are reasonable, believable observations, one from New Jersey and one from Connecticut, that I found online. I forget if it was INAT or Mushroom Observer. So the micro goes as follows. If you want to transition through the micro photos, this is a smash mount of some gill. So these elements were on the gill. I don't know if these are part of the gill or if the gill may have been pushed up against the veil at one point, and maybe some of the veil stuck to the gill. So this might be velar material, these globose sort of elements. But there were a lot of them, and then there were these things that looked like some kind of cystidia. That I found a few of these here and there on when I smash mounted the gill. I can't find any kind of detailed microscopic description of this Cystoderma aureum, or under the other name as well. Now, I just fished out David Aurora's book not long ago. He might actually have a description, so I haven't looked at that yet. Yeah, Mushrooms Demystified. So I'm going to have to read that. Now, I did get spores to drop from this thing, but not a substantial enough print to be able to assess pigmentation. So the spore print is supposed to be like a light yellowish, light brown yellowish kind of, but the spore print was pretty thin. I couldn't, and it was on a microscope slide, so I couldn't really tell if it was pigmented. These spores are not shaped exactly like what is reported for the species Pholiota aureum. They're supposed to be a little bit longer and a little bit thinner. So these spores are like 8 to 11 by like 5.5 to 7.5 for the most part. So they're a little bit thicker and but not as long. So the Q is different. But who knows how far back this description goes of Pholiota aureum. This might be 100-year-old description. I don't know. That's just been passed along for ages. Now, I looked at these spores under my better microscope, which I cannot get good pictures from because it's binocular. It doesn't have the camera input thing. And I'm pretty sure that what I saw was ornamentation on the spores, like some punctate kind of ornamentation, maybe really small warts. I'm going to take a slide with the spores on it over to a friend of mine's house tomorrow. He has a really nice microscope, and we're going to look at these spores at 1,000 power, and to see what they look like then. But anyway, Amanita spores are not ornamented, but Amanita spores do have a tendency to have a lot of like grainy contents inside them. And that could present the illusion of ornamentation. So, so I want to see what these spores look like under a better microscope at a stronger magnification. These photos are taken through my monocular scope. It's a good enough scope to get spore dimension estimates, but you don't see a lot of detail. These last two photos are Pileopellis, and so it looks like a sort of a cutis of chains of elements that are inflated in the midsections, let's say, and these look like terminal elements here, probably coming off of the top of the Pileopellis. I'm not really sure.
It looks, Dave, it looks, is that last photo, those things are coming on like a perpendicular? Or at
Not
least,
really perpendicular. They're just sort of, I'm not sure I would say perpendicular, because I know what you mean, because that would be in what's the name for that thing?
yeah.
Ixocutus or something.
Yeah, I don't know the name, but I know that's a distinction. Yeah.
Yeah, but I don't think these things. I think these are just, I tore off a piece of the Pileopellis and smashed it, and so some of these are just got smashed.
Gotcha. So
Yeah.
So not necessarily arranged as they were
No,
naturally.
I don't think there's any kind of perpendicular arrangement here. I think these are just elements that you can see. It's sort of, if you look down the bottom of the photo, you can see it's kind of gradual, in that the orientation of these things is, when you get closer to the top of the photo, they start to angle a little bit up toward the ones that are projecting. So I think that's what's going on here.
And also, because I know you're fighting the urge to call this thing an Amanita, or that's at least in the back of your mind. It doesn't
Sure.
read Amanita to me. But you know Amanitas more than I do. So,
what Amanita would it be? Which
Well,
are there Amanitas that have
I don't
yellow?
know.
Are there Amanitas that have yellow gills?
No, I don't think so. Okay, but there's a complicating factor here. Go back through the mushrooms. This mushroom these mushrooms were infected by some sort of bacteria or something. Now you see this in the pine barrens sometimes with the tricholomas, this like red infection. See on the bottom one there? So I don't know. Is that responsible for altering the appearance of this mushroom? Quite frankly, I think no. Okay, I mean this would be a big alteration. Look at these gills; they are completely yellow, really. Now the yellow might be a little bit overdone in the photo. I'm not sure, might because of the lighting. But this mushroom was pretty much yellow. These mushrooms are pretty much yellow or maybe orange, like the one on the bottom is a little more orange. That these mushrooms were yellow. And look at the context in the stipe is white. So
Right, like
you don't see any like skewing toward yellow, in the context. So
yeah,
the
you
contrast
mentioned
tells me these gills really are yellow.
you mentioned someone on iNaturalist called this an Amanita, and maybe that's why you're kind of chasing that a little bit.
Well, that's why I'm
But-
chasing it, right? Well, I think INAT actually probably called it Amanita because the guy
Right.
who was with me, he didn't know what it was.
He
And
probably
he just got
the
some photos and put it
AI
up.
probably thought it was Jacksonii,
Yeah.
Yeah, something like that, or
Which it-
flavoconia or something.
which
So
it-
why can't this be just a Amanita section of Validate? Are the gills free?
Well, yes, that's if it's an Amanita, it's probably Validae.
Yeah, and I
But
think
look
it's
at the way the
consistent.
partial veil transitions into a sheath. You can't see it here. You have to look on that one.
I don't think this is partial veil transitioning into the sheath.
No, not you can't see it on this photo. If you go to the other photo, not this one. These
Yes.
are old and beat up also. So this one here.
It's just the skin.
No, I think there's a transition there. I think there's a transition there.
Okay. This looks like an Amanita to me. There's something weird happening with it. Flavorubens, flavoconia, I don't know, but like late fruiting, weird stuff happens.
Right.
Are the
Well,
gil- are the, are
that's
the
why
gills
I'm not
free?
dismissing Amanita. And the spores actually, the spore dimensions fit in with Validae.
Yeah,
And they're
they're
pretty
elsewhere.
variable also. The spore dimensions are pretty variable, which you'll see in Amanitas. But I'd like to see the spores under better magnification with more detail because I want to see if they actually are ornamented. Because Amanita spores, to my understanding, is that they're not ornamented. They might have contents, but they're not ornamented on the surface. You can't tell from here, right?
Well, I'm still trying to get a notion of the gills. Are they free or not?
I think they're free.
Well, I think so too.
You know what? That doesn't even matter because a lot of Amanitas have attached gills anyway.
No, it's pretty rare.
It's not all that rare. I see it all the time.
The spore print would definitely be white, though, right? And so,
Yes, the spore print would be white for an Amanita, and that I was unable to assess. I got a spore print, but it's so
Right.
thin that I can't tell. It looks like it might be slightly pigmented, but it's on a microscope slide, and I took it outside. I put the slide on a white piece of paper, and I couldn't see any contrast. But
yeah.
it's a very thin print, so I'm saying that I can't tell.
Mhm.
But yeah, I agree. This could be an Amanita. I'm not saying it isn't. I don't
Yep.
think it is, but I'm not saying that it isn't.
This looks like a wart on the top of it right here.
Hmm.
And this
Oh.
one, do you see that?
Yeah, it's a beat up section of Validae. That's
Yeah,
what
it might be. It might be. But
I don't think
hopefully
this
I
is
can
just the
find
derma
out.
or the Theo, whatever it is, which is monotypic. We just have to look at the facts here.
Well, I'm sorry, what facts do we look at? I'm sorry, I missed something there.
Just the gestalt. We're looking at an Amanita.
Oh, well, the Gestalt is not different from this other thing.
Well, I think it is.
Look at some of the photos you find online. this thing
Pholiota
changes as it ages. It loses some of the,
Pholiota does not look like an Amanita, but this one does look like an Amanita.
yeah, it does. It does, but there are things that point away from Amanita. Yellow gills. I think the
I think it's
stalk
just
looks like it's sheathed on
it's
the
a leaf
one.
fruiting. There's something going on in there. There's no sheath. It's just the stipe tissue.
No, I think it's sheathed. It's sheathed, but it's old and it's weathered.
The flesh is white, non-staining. The
Yes, that's right.
veil is
And that other thing has white flesh.
very consistent with just an Amanita.
Yeah, but it's consistent. A lot of the traits are consistent with this Pholiota thing.
But the thing is, Pholiota does not look like an Amanita. This thing does look like an Amanita.
Well, I'll say
More
this.
warts
that,
over here. You can see
one
more
of the
warts
things
over
that's
here.
wrong for Pholiota is that
Yeah,
the
look at
cap
that.
is planar, and that species seems to not do that from the photos that I've looked at online. But this
Wow.
one's old. This one's old, and most of the photos you see are like taken of fruit bodies in prime condition.
Well, save some, Dave, and send it
Yeah,
out.
I'm going to pull it out of the dryer as soon as we're done here.
Yeah.
But the thing that's a complicating factor here is it looks like there's some sort of infection, so that may have accounted for atypical morphology.
Maybe. Yeah, interesting. Really nice to see the bright color, though.
Yeah. Yeah, the yellow is so rich on the gills. It's really interesting.
All right. That was number one. Let's keep going here.
All right.
Second one.
Oh,
Neolecta.
yeah. So I find this every year in the same spot, and it seems to be a very rare species. There are some photos you can find online that are labeled Neolecta vitellina, and you know what? When I searched online for photos, I'd say approximately 2/3 of them are incorrectly labeled. People have things like
It's geoglossum.
microglossum. They have microglossum
Yep.
identified as this. So this is now I know that this is Neolecta vitellina. I did a lot of micro on it, but that wasn't enough. I sent some to Linus. He sequenced it. It's 100% match for Neolecta vitellina on GenBank, submitted by David Hewitt, who's pretty much like the expert on North American Neolecta. There's only two species. There's vitellina and irregularis. Irregularis seems to be more common, but not around here. It's more of a northern species. It's if you go into the Adirondacks, you have a pretty good chance of finding it. But this comes out every year in the same spot. And I used to think it was Scots pine because there are some Scots pine in this area. But I had a tree expert with me yesterday, and he pointed out that the pines that are nearby where these things are growing are Virginia pine and pitch pine. And some of the bigger ones he thinks are Virginia pine. So anyway, seems to be a pretty rare species. And it seems like it's probably mycorrhizal because it's in the same spots every year. Comes out late. Usually you don't see it before November. And then a warm fall, you'll see it all the way into late December in this one little area. Maybe 10 or 20 square meters, something like that. It spreads out, around the bases, not the bases, but near where these pines are on the ground. So that's an interesting one.
Yeah, I love that. I found a Neolecta before. I'm not sure which one it was, though. I'll have to look back.
Hey, Neil, oh, you did? Yeah.
Yeah,
That
yeah.
start was here. So was it more like weirdly shaped?
It was pretty similar to this.
Well, maybe it was vitellina then, because the other one, irregularis, that one is more like chaotically shaped. You know, these are kind of like nice clubs, spatulas or clubs or whatever, you know?
Yeah, I love the look of the two that are kind of twisted. Yeah, yeah,
Yeah.
that's really cool. Yeah, I'll have to look back. I don't remember, but
Yeah, chances are you found this then.
yeah,
If you found
maybe
ones that are nice club shaped ones.
I'll have to take a look.
Dave, when you were going through them and you said 2/3 of them are probably misidentified under this name,
That's what I've seen online. Yeah.
were some of the other names trying to call them oh god, what is it, cordyceps?
Yeah, cordyceps. I think there were one or two cordyceps that were called this. Mostly it was it was microglossum.
Yeah, that microglossum, what is it, lutescens,
Yeah, I
probably
forget, but it's the yellow one. You know, it's the one
the
that
yellow
looks.
one. Yeah.
So on my so up on our property in our upper yard, we have this place where sometimes we'll have like a bonfire. So we'll stack up some pieces of wood that we cut. We had a bunch of ash trees die over the last 15 years, so some of those we've been cutting down, and we just stacked the wood. And so this was on hardwood, maybe ash. It was hard to tell. It's getting decomposed. So it's Resupinatus. It's these tiny little gilled mushrooms that look kind of like Crepidotus or something. They're very small. I'm pretty sure this is Resupinatus. I did get a few spores to fall, very few. But I'm pretty sure this is Resupinatus. I haven't yet attached a species name to this, but I think I probably can as soon as I just kind of look up. Because I did get a few spores, and I think they are. It looks to me like they're ellipsoid, and I think some of the species of Resupinatus have like globose spores. This is taken through a loop. This photo.
That's great. Great
Yeah,
shot.
it came out pretty
Yeah,
good.
that's a
Sometimes
good
you get
photo.
lucky. So,
Beautiful.
yeah, and
Yeah,
so there's not many spores to see there. There's like 2 spores, I think, because not many fill. But I think that the fact that they are ellipsoid is going to lead to one species rather than the other several.
Dave, when you take a picture through a loop, do you put the loop down on something, or I mean, are you holding both in your hands?
Yeah, I hold the camera and the loop, and I have to position the subject, you know,
Mhm.
so that I'm able to aim the loop and the camera at it.
Yeah.
But sometimes it doesn't work very well. This time I got lucky. I only took a few photos. I had a few other photos through the loop as well, and they weren't terrible. But this was the best one, so I kept it.
Yeah, it's hard to do for sure. That's beautiful.
I love these Resupinatus. I see them on firewood a lot as well.
Cool.
They're beautiful.
Yeah, so you get an idea how big these things are. They're pretty
Yeah.
small.
They are not big. Yeah.
Those are the upper parts of the caps,
Yeah.
upper surfaces.
I put Resupinatus applicatus, applicatus, whatever it is.
Yeah, I
in
think
the
that's the
chat.
one it is.
Uh,
Is
I
that the
meant to
one with
just
the
put.
ellipsoid spores?
Honestly, I forget. There are a few species that was just on my brain, and I put that in the chat. I'm not sure if that's what it is, but yeah.
Yeah, I only got around to creating the observation like 20 minutes before we started. So I've just got my photos all straightened out, late this afternoon. So I haven't really researched the species names yet. But I know that there's one of these Resupinatus that has ellipsoid spores. So I'm probably going to be able to attach a species name to this.
Cool.
Yeah, that
Well,
was a good one. Good one to
yeah.
find.
Like to see that. All right. So what was that? 2? Is this?
No, that was 3
That
actually.
was 3. Okay. All right.
Yeah.
All right. So we're at 4 now. Okay.
Yeah, we'll go quickly. Okay, so Igor's here, right? Igor, what do you think? Xerocomus spadiceus. This is the thing I used to call Xerocomus subtomentosus, but that's not something that a name we use anymore in North America. So I got this name off Michael Quebec.
Yeah, I don't know. This genus needs a lot of molecular work and
Yeah,
elucidation
they even say so on
and
Michael
typing.
Quebec.
I just got 2 Xerocomus sequenced, and they are in pherogenous group. But if you blast them, you get Ernst both collections called spadiceum, and there are all these other names. It's just ridiculous.
Well, that Michael Quebec sort of hints around at the same thing you're saying. But this seems to be the best available name right now. And it looks like this one was kind of old. Looks like it's, that brownish color inside this stipe. That's probably a sign of pre-decay. And this stain on the bruise on the pores, it took a long time for me to see it. I scratched them and nothing happened. And I looked a couple minutes later, nothing happened. And then hours later, it had darkened to this extent. So I'm not sure what to say about this bruise, but it did change. It did change color, and there's a tiny bit of bluishness in there, I guess. But yeah, I'm going to call this thing Xerocomus spadiceus for lack of better name.
Xerocomoid. If you hear people talk about the xerocomoid, it refers to this kind of weird thing going on with the pores, right?
They are irregular, and they don't have the glue that separates the tubes. Like unlike other bolids, like the bolitoid type of hymenophore, these tubes just rip apart. You just rip apart the tube. They're all glued together. I mean, they're basically not. There's no glue. I mean, they're all together. So
Yeah.
that's kind of a definition of xerocomoid. But even things which are in boletoidae, like xerocamellus, sort of have xerocomoid pore surfaces. So
Yeah.
this thing is not really applicable to xerocomus alone, or
Yeah.
anything from xerocomoidae.
Yeah. But that xerocomoid look to it. Yeah.
Yeah, they are irregular, angular, and the tubes cannot be separated from each other. They just are
Rip.
torn apart.
Yeah.
Yeah, no staining on the flesh. So
Yeah, the Coma collection that came from coniferous forest and the one from Pennsylvania when I visited you in July in deciduous woods, they had the same DNA. So I'm not sure what to make of that.
Okay.
And they didn't stain blue on the inside either. But the pores stained blue, at least in one of them. Yeah, it's in the pherogenous group. That would be the more accurate, probably.
Pherogenous group.
Yeah,
Yeah, that
circumscription. Yeah.
Doesn't that one have more ornamentation on the stipe though?
Well, perhaps, but I find these things somewhat variable. All of Xerocomus. So yeah, it's a cryptic genus. It's not a big one, but I haven't seen any publications to elucidate, other than the molecular work by Colin Damnauer, who sequenced type collections from Michigan. So we know what Tanex is genetically now, and we have more than one. But yeah,
there's more than one 10x.
Yes. Yeah, there's more than one 10x.
Well, that's interesting because that one's actually fairly distinctive, visually.
I think we're just finding out there's like a duplicate of or triplicate of everything.
Yeah,
There's
yeah.
more than one in Nixus, Auricularia in Nixus. There's more than one Auricularia Roxani. So
Uh-huh.
yeah,
Yeah,
I-
I'm actually not surprised about either of those.
yeah. Well, we have DNA data to support that, and
Right.
we just have to wait for the dust to settle and then work through it.
Yep.
Yeah, Xerocomus is just yeah. I just shrug my shoulders and that's it. Call it what you like.
Call it what you like. Okay, well, I'll call it spadiceus. Okay, so INAT does not allow for proposing the name Antosibe priscua. And I think that's what this is. So it's a name found on Myco, Quebec of a, a small genus, Antosibe, in the Entolomataceae. The spore print is pinkish. This thing, I've been finding these under pine. This isn't the only one I found. I found a few others under pine in other locations. And the spore print is pigmented. It's, a little bit pinkish. And here's the kicker. See, it's the spores. And I don't know if you can, how much you can enlarge this.
That's it.
Not that much, I guess. The spores are vaguely angular. They're not
Yeah.
like strongly angular like true Entoloma spores. They're just vaguely angular, and when they're not profiled favorably, they don't really look very angular at all. Sometimes they look like they're just flat on one side. But that fits. If you look online, you'll find some photos of Antosibe spores, and also if you look on Myco, Quebec, for the description and the portrayal of Antosibe priscua, this fits pretty well. Now it grows under pine, in pine needles, has these vaguely angular subglobospores. So I think that's what I've got here. And I naturally see on Mushroom Observer, you can, create a name. Anybody can create a name, and back it up with as much reference as possible. And you don't even have to do that. But on INAT, you can't do that, unless you're like someone deigned with the authority to do so. And I don't know how to achieve that. But so the best I could do on INAT was Entolomataceae, because they wanted to call it Nitida. And Nitida apparently is a section of Entoloma in some people's eyes. In other people's eyes, Nitida is a species, and sometimes it's also called Nitidum. So there's Entoloma nitida, Entoloma nitidum. I think they're the same thing. I'm not sure. But this is not it. This is definitely not that thing. So I'm not going to go with INAT's recommendation. I'm just going to call this thing Entolomataceae, and I put into the notes that I think it should be called Antosibe priscua or Pishua. So I've got this. I think I may have sent one of these in to Michael Blitz. So maybe we'll find something out. What's the date on this one? This one may have been sent in
November 2nd.
No, too late. Okay, but I think I found some of these earlier. I think there was one that I found during Myco Blitz, so I was able to send it in.
Good.
So yeah, hopefully we'll find something out.
Okay. All right. Well, that brings us up. We're just 6 minutes past 9. I'm going to stop the recording here. Find a way to do that. So thanks everybody for contributing. Good session.
All